We're recording at this point, and I just want to let everyone know who's here that if you have questions throughout the presentation, I want you to put your questions into the Q&A down at the bottom of the screen. You'll be able to see that. And Dr. Fajgenbaum will take opportunity to pause at certain points along and make sure that you get your questions answered. If you mistakenly put a question in the chat, we'll try and get to your question in the chat as well, but it's a little bit easier to keep track of the questions in the Q&A function. So it is my great pleasure to introduce to you David Fajgenbaum. David is a graduate of Georgetown University, the University of Oxford, the University of Pennsylvania, and the Wharton School. An assistant professor of medicine at the University of Pennsylvania and the associate director for the Orphan Disease Center. He is co-founder and executive director of the Castleman Disease Collaborative Network, as well as the co-founder of the National Students of Ailing Mothers and Fathers Support Network. Dr. Fajgenbaum has been recognized with multiple awards, including the Forbes 30 Under 30, for healthcare and the Rare Companion of Hope Award for Science. He has been profiled by major media outlets such as the New York Times, Forbes, Science, Reader's Digest, and the Today Show. And he lives in Philadelphia with his wife, Caitlin, and their daughter, Amelia. So thank you very much. We're pleased to have you.
Well, thank you so much, Jackie. It's really a pleasure and an honor to chat with you all today. As I was sharing earlier, I've had the good fortune of spending a lot of time interacting with post-bac students, pre-health students at Penn, both as work-study students in my lab, also as employees, and I've just been so impressed by all of you and your colleagues. Begin sharing my screen. Great. So I titled my talk Chasing My Cure, and it's because like so many of you, I set out chasing after cures. I wanted to become a doctor, and I wanted to take care of patients and chase after cures for my patients, when out of nowhere, I became deathly ill and began starting to chase after my own cure. And so during my talk today, I want to share a few different perspectives that I think are going to be important for you all as you think about your careers in health fields that I think will really resonate with you. And I believe most of you already got a copy of my book, Chasing My Cure, and I hope you'll enjoy it and that it will bring some inspiration to you throughout your journeys. So the 5 perspectives that I'll be touching on, the first is that of a caregiver of a patient. My mom was diagnosed with cancer while I was a freshman in college at Georgetown with brain cancer, glioblastoma. And she passed away about a year later. And watching her battle with glioblastoma, watching the way that she fought with so much strength and the way that her doctors took care of her really inspired me and motivated me to want to go into medicine. As I shared, I came to Penn for medical school. And in my 3rd year, I went from being a totally healthy medical student to dying in the intensive care unit, literally actively dying because my liver, kidneys, and bone marrow were all shutting down for an unknown cause. The same hospital I've been treating patients at. And then when I survived, I realized that I wouldn't survive long-term unless I got involved in research and began searching for a treatment that could maybe save my life. And then realized that it wasn't something I could do on my own, that I would need to become an advocate, and I would need to connect physicians, researchers, and patients together to take on this disease. So I'll be sharing these 5 perspectives: that of the patient, the physician, the advocate, the researcher, and the loved one of patients, the role that external bodies such as the FDA and pharmaceutical companies really all play when you bring these 5 perspectives together to make progress for individual patients, but also how important it is to bring all these perspectives together within a given disease, and then also sharing perspectives across diseases. So today I'm going to share my personal journey. I'm going to transition into an example of personalized medicine. I'm sure you've heard that term before. I'll define it a bit more, share some applications beyond Castleman disease, and then close with lessons learned. Just as was mentioned earlier, please feel free to enter in questions into the chat or into the Q&A. And we'll be sure that throughout the course of the talk, I'll take breaks to check those out. And I'd love to answer any and all questions that come to your mind. So as I shared earlier, I was a healthy third year medical student at Penn when out of nowhere, I began experiencing multi-organ failure. My liver, my kidneys, my bone marrow, my heart, and my lungs all began to shut down. And I was hospitalized at HUP, the hospital at UPenn, where I gained 70 pounds of fluid. I had a retinal hemorrhage that caused me to become temporarily blind in my left eye. I needed daily transfusions of red blood cells and platelets to keep me alive. And I drifted in and out of consciousness for weeks and weeks, all with no diagnosis. It was 11 weeks into my journey before I was finally diagnosed with what's called idiopathic multicentric Castleman disease. But I was so sick right before my diagnosis that my doctors told my family that I was unlikely to survive. And my family brought in a priest to administer my last rites to me. And so I've really considered that moment when a priest was in my room administering my last rites to me to be the start of my overtime, which is time I didn't think that I would have, but time that I really wanted to make the most of. And so this is a quote from my book: "The experience of being in overtime is surprisingly one of intense awareness and scrutiny and clarity. There's a reason for that. When there are only a few seconds left on the board, all distractions disappear and the purpose, victory, becomes clear. The present is only the things around you, and overtime is all present, nothing but present and purpose." And I knew a little bit about overtime because I played college football at Georgetown, and we actually lost a lot more games than we won or that we went into overtime. But I certainly did appreciate the sense of overtime, where really you want to make the most of every second. You can make a mistake in the first quarter and make up for it, but a mistake in overtime, the game's over. And that's really how I've been living, just trying to make the most of every second. Also during undergrad, I met my girlfriend, Caitlin, and we dated for a few years while I was a medical student, and actually broke up just a few months before I became so ill. And when we broke up, we both felt that we had all the time in the world. Here we were 25 years old. If it's meant to be, it's meant to be. And then out of nowhere, there I was dying in the ICU and realizing that I wouldn't have any more time. And I really regretted that I didn't make the most of the time that I had, and that I didn't fight for our relationship. And so this quote I want to mention, it came from the simple realization that the regrets that ate at me the most as I prepared to die weren't about anything I'd done. My greatest regrets were things that I'd thought of doing but never did. I wanted us to be together, but I knew I had, or I thought I had all the time in the world, and I didn't take that action. So I promised myself that if I were to get out of the hospital, I wouldn't do that again. So thankfully, right around the time of the diagnosis and having my last rites read, I was started on chemotherapy, a combination of 7 chemotherapies, because idiopathic multicentric Castleman disease, idiopathic means we don't know the cause. It may be a lymphoma, it may be an autoimmune condition, it may be triggered by some sort of virus, but we don't know. And so we don't know the cause of it, which makes it really hard to treat, but we do know that if you obliterate the immune system with chemotherapy, that you can treat it. And so I got a combination of 7 different chemotherapy agents. You can see here me with my dad, just so thankful that I'm alive. And this is a picture from a few days later. I'm now about almost 6 months into my battle on New Year's Eve of 2010. And I share this story in my book, and I'll share the anecdote with you guys now. And that's that on New Year's Eve, we went for a walk around the hematology oncology unit, and we passed the family waiting room, and we saw there was a gentleman who was kind of like swaying in his chair. He was visibly really drunk on New Year's Eve. And in our next lap around the unit, we saw that he had fallen onto the ground. And so my dad ran over to him and helped him back into his chair. And he looked at my dad and I, and he said, "Thanks so much. Good luck to you and your wife." We were really confused at first. What's he mean, wife? And then I looked at my belly and I realized that he thought that I was my dad's pregnant wife, which was a low point emotionally for both of us. But I turned to my dad. I said, "Man, Dad, you've got an ugly wife." And we managed to laugh really, really hard about this. So facing my horrible moments with laughter was just as fundamentally a rejection of Castleman's dominion over me as anything else I was doing. It cleared my mind, it stiffened my resolve. It was entirely up to me to determine what was and what wasn't funny. Perhaps most important, humor is social. For me and my family, there was never a better way to reset our collective resolve than laughing together. About 7 weeks later, I was discharged from the hospital, and you can see how big my belly was because my liver and my kidneys weren't working. So I was gaining fluid all over my body, but in particular in my abdomen. And you can maybe see how some of confused me as my father's pregnant wife. I'm bald. I've got this big belly. And then this picture is from a few years before. I mentioned I played college football at Georgetown, and I always say this is like the worst before and after picture of all time. But if we could flip the order, then I feel like this could be a great advertisement for Muscle Milk or for Peloton. But unfortunately, the pictures are in the wrong order. The one on the left was from a few years before the photograph on the right. So once I was out of the hospital, I took a picture every week for the next 8 weeks, and you can see my hair starting to grow back. You can see the fluid going away in my belly over the course of the weeks. And I took pictures from the side as well, and you can see the fluid going away and my hair starting to grow back. So when I got out of the hospital, the first thing I did was reach out to Caitlin, and I wanted to see if she felt the same way that I did, that regretting that we didn't try to make it work. And thankfully, she did feel the same way that I did, and so we started dating again. This is the first Halloween after we started dating again. This is me dressed as Borat, which I hope everyone watching this knows who Borat is. If you don't know who Borat is, I hope you'll look up who Borat is and be excited to hear that Borat 2 is coming out on October 23rd. And I hope you guys will all watch it. I'm a huge Borat fan. And this is a picture of Caitlin and I. This is, I think, a really good symbol of our relationship. So on one shoulder, I was getting my infusion. So when I left the hospital, they had me get an infusion every 3 weeks of an experimental drug, hoping that it would keep me in remission. And I'm getting that experimental drug infused into me on one shoulder, and then I'm leaning on Caitlin on the other shoulder. I think this is a nice symbol for how things were at the time. So I came back to Penn for medical school. Now, finishing out my third year, and a major donation was given to Penn to start an orphan disease center. Orphan disease is another term to describe rare diseases. It's called orphan because they're rare and neglected. And so the Orphan Disease Center was started, and I immediately asked to be involved in ODC and helped out with early strategic planning for the center. When about a year later, I relapsed. I nearly died now for the fourth time. And this time I now relapsed on the only drug in development. And so it wasn't just that there were other options. I now literally had run out of all options. And my doctor explained that to me. And so I promised my dad, my sisters, and Caitlin that I would dedicate my life to trying to find a treatment for my disease. And I had conducted a little bit of research before this point, but I certainly was not very experienced performing research, but I knew that it was really my only shot. So I began conducting laboratory work at Penn. I also started a foundation called the Castleman Disease Collaborative Network to bring together physicians, researchers, and patients from around the world to really accelerate research on a global scale. This was a really important moment in my life. I'm literally alive today and sitting here because of the moment where I went from being a very hopeful person who just kind of believed that if I hoped that someone would do the research, and if I was a positive person, that someone would find a drug that could help me, to realizing that if I'm hoping for a treatment, then I need to turn my hope into action. So hope cannot be a passive concept. It's a choice and a force. Hoping for something takes more than casting out a wish to the universe and waiting for it to occur. Hope should inspire action. And when it does inspire action in medicine and science, that hope can become a reality beyond your wildest dreams. So a little bit about Castleman disease and where we were back in 2012. There was about $10,000 a year going towards translational research. Translational research being research to identify new drugs, new diagnostics, but really making progress, translating basic science findings into clinical implications. So that's basically no money. There was no funding from the NIH. There were no patients in a registry because there was no registry. There were 2 advocacy organizations, and then we became the third, but there was no sort of unified research plan. There were no cell lines or animal models, registries, or biobanks. A lot of questions remained. There was no diagnostic criteria to help to go through a checklist to diagnose patients with IMCD, no treatment guidelines, no predictive biomarkers, so no tests you could run to predict whether someone would benefit or not from treatment, no FDA-approved drugs. And then the one drug that was in development, siltuximab, didn't work for me. So it was a frightening feeling to get out of the hospital and to see how weak the field was and how much work was needed, and how, frankly, unlikely it was that we would make progress in time to save my life. So a little bit about Castleman disease. I like to try to pull complex concepts together into a pretty straightforward diagram, but the disease is actually quite complicated. So at the heart of Castleman disease is a cytokine storm. You've probably heard the term cytokine storm a lot lately with COVID-19. Cytokine storm occurs when your immune system is out of control and your immune cells produce cytokines. Cytokines are just messengers or molecules your immune system uses to communicate with other immune cells and to fight off invaders. And so you have a cytokine storm, the immune system's out of control, you have a particular appearance to your lymph nodes, and you feel really, really, really sick. Your liver shuts down, kidneys shut down, bone marrow. That's kind of what we know about the disease. It wasn't until the 90s that interleukin-6, which is one particular cytokine, was discovered as being important as really causing the disease in at least a portion of patients. But even still, there was a black box around, well, why is interleukin-6 elevated in the first place? We didn't know what intracellular pathways were involved, what cell types were involved, or even what was causing the cytokine storm in the first place. Unfortunately, we still don't know what causes the cytokine storm, but we do at least know more today about why the cytokine storm happens. So at this stage, we knew interleukin-6 was important, and we knew that if you blocked interleukin-6, that a third of people would get better. Everything would resolve if you block interleukin-6 with a drug called siltuximab. But unfortunately, for 2/3 of people, blocking IL-6 with siltuximab is not effective. And so what do you do for people where interleukin-6 blockade doesn't work? And chemotherapy was really the only option. And so we decided that we wanted to focus on trying to identify new therapies for people who don't benefit from IL-6 blockade. I should mention that IL-6 was discovered as being important by Kazu Yoshizaki, photographed here on the screen. Kazu discovered interleukin-6 was elevated. He developed a drug to block interleukin-6 signaling. And I heard a rumor that he tested it on himself. He gave it to himself. And when I asked Kazu, I said, Kazu, Bakoto just told me you tested Tosi on yourself. He said, no, I didn't give it to myself. The nurse, she gave it to me. So he made the discovery, he developed the drug, and he also gave the drug to himself. Amazingly, about 20 years later, and by the way, it went on to get approval in Japan for Castleman's, and then it came to the United States and it's approved for rheumatoid arthritis. But about 10 years ago, the first pediatric patient received something called CART therapy. It's a cellular immune therapy to treat cancer. And this young patient, Emily Whitehead, photographed on the screen, was dying in the intensive care unit with a syndrome that looked almost identical to Castleman disease. And the doctors decided to try tocilizumab to treat her. This drug Kazu developed years ago for Castleman's, and it saved her life. And since then, it's become standard of care for patients who get CART therapy. More recently, it has been used extensively for COVID-19. And actually, there have been a number of clinical trials that have shown both promising but also less promising results related to the use of this drug. But I think it's exciting when you can see a drug like this developed for Castleman disease, which is a rare condition. Only about 5,000 patients are diagnosed each year in the US. So it's about as common as ALS. You've probably all heard of ALS. You probably have not heard of Castleman disease. But with that in mind, it's a rare condition, but certainly there is major unmet need. Just for time's sake, I'm actually going to skip through this pretty quickly, but it's basically to say that we've taken a new approach to research. The traditional approach to research was to raise money and then invite researchers to apply to the foundation for funding to do the research they wanted to do. So a small fraction would apply for funding and an even smaller fraction would receive that funding. The research would be done, and then 6 months later or a year later, you do it all over again. That's a traditional approach to research. But basically, in this approach, you're hoping that the right applicant applies for the right project at the right time. And it's really unlikely when you have a small number of applicants that all the stars will align. So we created what we call the collaborative network approach, where we first built a large community of physicians, researchers, and patients. Then we crowdsourced from the entire community. We asked the entire community what research should be done. Once we prioritized that, then we said, who is the best person in the world to do that research, to do the things that we've now prioritized, completely flipping the model on its head. Next, we went out and we raised the money for that project, for that researcher. We got the samples in place for that project. We execute the study. We look for drugs that can be repurposed. We share that knowledge with the community, and then we just keep crowdsourcing over and over again. This approach leverages the whole community for ideas and make sure that we're matching the best ideas to the best researchers as quickly as possible. I knew I couldn't do this alone when I came up with that plan and that idea. So I started the CDCN, like I mentioned, and I held a webinar with a bunch of Castleman's patients, and I showed this image of a Castleman. The disease is named after a doctor, Benjamin Castleman, but we've kind of repurposed the idea or the name Castleman to be a castle man, like a warrior. And we showed this picture to a bunch of patients, and then this showed up on our Facebook page. So one of our patients screenshotted it and got it tattooed on his shoulder. And today there are literally hundreds of Castleman tattoos all over the world, which I think is just a representation of how excited our patients were to get behind our community. I was a medical student when I was first diagnosed, and then I was a business school student in the midst of building out the CDCN, both at Penn, and I was able to enlist a large number of my classmates, medical students, business students, as I mentioned, some post-bac students who really got involved in various capacities as volunteers, and this is us doing what we call the Castleman Flex, showing that we're taking on Castleman disease. We built a huge network of physicians and researchers from around the world. And really, it was only through this community that we've been able to make the progress that we have. So I'm going to take this as a moment to take a look at the chat function to see if there's any comments or Q&A that have come in. Let's see.
No, there's nothing in the Q&A.
Great. So as I said earlier, feel free to throw in your questions or comments if they come up, and I'll jump back into the presentation. Great. So you all have probably all heard of the term personalized medicine. Personalized medicine is this idea that rather than treating everyone with a condition the same way, that you use information to personalize treatment to that particular patient. And so let me show you a visual example. So the traditional way of doing medicine is you've got a group of 6 patients with a given disease. You give all 6 of them drug A. So for Castleman disease, this would be siltuximab. And then out of the 6 people, 2 patients benefit. There's 2 responders. And then the other 4 patients don't benefit from that drug. And so the next thing you do is say, well, what's the next drug we can try to give to the non-responders? And then hopefully out of the non-responders, you'll get 2 people who will benefit and the other ones won't, and you'll just keep this cycle going. But personalized medicine is the idea that you start out with the exact same 6 patients, but the first thing you do isn't give the same drug to all 6 of them. It's to run a series of tests, whatever they may be, to determine who should get drug A. And you say, well, the people who are going to benefit or likely to benefit should be the only ones who get drug A. And then you also identify additional drugs to give to the patients that would not have benefited from A and would benefit from B or would benefit from C drug. And so the idea is to use, oftentimes it's omic data, genomic, proteomic data to figure out who is likely to benefit from what drug, and can I run those tests ahead of time so I can make sure I get the right drug to the right patient at the right time. It also requires identifying new drugs, because if you just have one drug, personalized medicine isn't very helpful because you don't have any additional options for people unlikely to benefit from that drug. So I began trying to practice this as I finished up medical school. This is a picture of Caitlin and I on my graduation day. And then this is a picture of us a few months later when I relapsed. So now for the 5th time, I nearly died from Castleman disease. You can see I'm bald from the chemotherapy. And all I wanted to do was make it to May 24th, 2014. That was our wedding date. And I so badly wanted to make it to our wedding date. But I knew that I wouldn't if I didn't identify a drug that could help me. So what do you do when you've run out of options? So all drugs that had ever been tried for Castleman disease have been tried on me. Well, you have to take a novel approach, come up with a new target. And so I went through data that I'd been generating on my own samples for the last several months. And I identified a couple trends in my data. The first was that I found that there were specific cytokines. I explained what cytokines were earlier, but specific cytokines that began to rise in the weeks leading up to my previous relapse. So all the cytokines were elevated during relapse, but what cytokines began to rise beforehand that could maybe suggest that they're involved in causing Castleman disease or driving Castleman disease? And I found that 2 particular cytokines, one's called VEGF, vascular endothelial growth factor, and the other one is soluble IL-2 receptor, which is a marker of T cell activation. So I went back to samples I'd banked on myself, and I confirmed that in fact there were increased levels of both VEGF and also activated T cells. Then I looked at my clinical data, and I found that there were a lot of clinical correlates. So VEGF causes blood vessel growth. Well, my lymph node had incredible amounts of blood vessels all throughout it. My skin had what are called cherry hemangiomas, which are literally blood vessels growing on my skin. And the incredible fluid accumulation I had is often seen when you have high VEGF. So this is an example where the laboratory work I was doing fit with the clinical picture of what I was showing and what I was presenting with. And so we asked and we wondered, is there something involved in the cytokine production we're seeing, the T cell activation, and the VEGF production? Is there a common pathway involved in all three of these? And so we ran the data through what are called pathway analyses tools, and the tools predicted that the mTOR pathway, mTOR is an important communication line in the immune system that helps immune cells to communicate with one another and also fight off infections. And we found that the mTOR pathway was predicted to be highly activated. So we went to the tissue to really prove this. Is it true that mTOR really is active for me? And so the first thing you do anytime you do research is you have a comparator group, a normal group. And so we looked at normal lymph nodes, and brown is positive, blue is negative. And we see in a normal lymph node that there's some mTOR activation. You can see there's some cells that have increased mTOR activation. Then we looked at my lymph node and we were just blown away. A tremendous amount of mTOR activation, really off the charts levels of mTOR. So at this stage, this was really exciting because there are drugs that are already FDA approved that block the mTOR pathway. And so from this, we said, okay, well, maybe we should start testing an already FDA approved drug like sirolimus on me to try to inhibit mTOR. And maybe by inhibiting mTOR, we can stop this disease in its tracks. And so I started taking sirolimus 81.29 months ago that I've been in remission. And I say 81.29 months because I know that I can't round up. I don't know if I'm going to make it to 82 months in remission on this drug, but I also refuse to round down because my team and I worked so hard for every fraction of every month that I'm just so grateful for it. The New York Times called this "Doctor Cure Thyself," which I think is a bit of an overstatement. I think it should be doctor helping himself a little bit right now and hopefully for a lot longer. But for now, I'm just so thankful that it has been helping me. And this is a picture of Caitlin and I on our wedding day. So I made it to our wedding day, and you can see my hair actually grew back just in time for our wedding day. And 2 years ago,
we do have a question if you want
great.
to check the Q&A.
I'll switch over to the Q&A and take a peek at it. Let's see, when does the time necessary to run these tests come into conflict with the urgency of someone's condition? What do you prioritize? So this is a great question, Dean. So you always prioritize the condition. You run tests around the condition. You make sure that you're doing every treatment, you're doing everything you can to protect and take care of the patient. And anytime you get a chance to collect a blood sample for research or to do a research study, that's great, but you never let research get in the way of clinical care. In my case, I shared about how we found these things from my 5th relapse, but we didn't find them while the relapse was happening. We found them by, once I survived that relapse, looking back at all the data, looking back at the samples we had banked, and then doing the work on those samples. Because to your point, it's almost impossible to do this in real time, and you would likely get in the way of patient care if you're too busy doing research. And so really you want to do research when you can, and as soon as you have the insights from that research, that's when you want to then make sure that you can apply it to your clinical care. That was a really great question. So then how are resources distributed when needing to run scientific tests for an individual to receive specialized drugs? It's a great question. There is no resource distribution, or there is no sort of overarching group that decides which tests get run on which people. That doesn't exist. It's something that every researcher, every physician can decide what tests they want to run and don't want to run. Generally, whether it's going to be covered by insurance is a big part of whether to run the test. If it's not covered by insurance, it needs to be covered by someone, either the patient or the research lab. So there is no sort of overarching, you can do this research or that research. It gets approved by the IRB early on, but not on a case-by-case basis. And then there's a good question about COVID. I'm actually going to jump back into the slides because we're going to get to COVID in just a bit. But please do continue to enter in your questions into the Q&A part. So a couple of years ago, in the midst of this really long remission, my wife and I had our first daughter, our first child. This is Amelia, photographed here with Caitlin and me about a year ago. And this is Amelia on her 2nd birthday just a couple months ago. And just really, for me, hard to put into words what it's meant that I've been able to survive, get married to Caitlin, and be able to have this sweet daughter, Amelia. So beyond my personal journey and beyond my own personalized medicine, we've thought a lot about how this affects other Castleman's patients. Last year, we published the first description of mTOR as a therapeutic target in Castleman's and the use of sirolimus in 3 patients. This year, we published applying this to 26 Castleman's patients and how we find mTOR activation quite uniform across patients. And also this year, publishing about a potential mechanism for why the mTOR pathway may be activated in Castleman disease. This research has led us to try sirolimus in more patients. This is Joey, who showed up to the Children's Hospital of Philadelphia right next door to us about a year and a half ago. And he wasn't responding to any therapies. And so we tried sirolimus. He had a really, really positive response. Unfortunately, out of the about 30 or so patients we treated with sirolimus, only about 1/3 to 1/2 of them have benefited, and the other about 1/2 to 2/3 have not benefited. There are 2 patients in particular, Lisa and Sergio, who I think about a lot because they were given all lines of therapy, including sirolimus, and unfortunately nothing could stop their disease, and they passed away battling Castleman disease. And they really inspire me every day to continue to fight to push forward the science for patients like them. So thinking back to our figure and where things fit in, I did want to highlight one research study we did that tries to get to this idea of what tests can be done to determine who should get one drug or another drug, and what other drugs could we potentially discover. So we did something called serum proteomics. We took blood samples from almost 100 Castleman's patients, and we measured 1,300 different proteins in the blood of each one of those samples. We also got samples from related conditions in healthy individuals, really with the goal of saying, if we measure 1,300 different things in the blood of Castleman's and other diseases, we can start to figure out which of those 1,300 things maybe could be diagnostic tests to say someone has Castleman's or not, or maybe could predict if someone gets better with one drug or another, or maybe even help us identify things that we should direct drugs against, what we call therapeutic targets. And the first thing we did with our data is what's called a Tisney plot. Some of you all may be familiar with this, but Tisney plots are a way to cluster samples together. So there is no X and Y axis. Basically in here, every sample is located in the position, every icon represents one patient sample, and every sample is where it is relative to the other samples. So if 2 patients have almost identical levels of 1,300 different proteins, then they're going to be really close to one another. But if 2 patients have very different profiles, so if they've got high IL-6 versus low IL-6, but differences across those 1,300 tests, their samples will be far apart on this Tisney plot. They will not cluster together. So the closer 2 samples are together, the more similar their proteomic profile is, so the proteins in their blood. And so we did this with related diseases. The lymphoma samples clustered together really, really well. The HIV positive, HHV8 positive samples clustered pretty well together. The rheumatoid arthritis samples also clustered well together, suggesting that their proteomic profiles are very, very similar. And so we wondered, where is Castleman's going to fit on this? Where is it going to cluster? Is it going to be more like an autoimmune condition, more like an infection, more like a cancer? And what we found is that actually it clusters all across. And so it doesn't even really cluster. It's spread all across the Tisney plot. So you have some patients where their proteomic profile is almost indistinguishable from lymphoma, and other patients where the profile is almost indistinguishable from rheumatoid arthritis. I'm really highlighting this is a disease that spans the nexus of autoimmunity, cancer, and infectious diseases. So we use that data to then say, well, are there particular subtypes within Castleman disease that have meaningful clinical associations? And we found this one cluster in particular, cluster 1, that had a significantly improved response to siltuximab. So though only 1/3 of all people will benefit if you give them siltuximab, it was closer to 2/3 of people within the cluster 1 group would actually benefit from siltuximab. The reason this is important is because we could actually take the 1,300 proteins that help us to cluster people into cluster 1 and identify the 7 proteins that were the best at doing this. And we found that just measuring 7 proteins, we could determine who's likely to be cluster 1 versus not. And then we looked in a totally separate cohort of patients, and we were able to validate this finding with cluster 1 patients being more likely to benefit than not benefit. So now we have a test, like we talked about earlier, a test where you can say, if you have this profile, you should get siltuximab. If you don't have this profile on these 7 proteins, then you shouldn't get siltuximab. Well, what do we do for the people who shouldn't get siltuximab? What else can we target? So there's something called the JAK-STAT signaling pathway. JAK-STAT is activated in the presence of interleukin-6 and other cytokines. And importantly, we found that IL-6 JAK-STAT was highlighted as potentially being involved in these cluster 1 responders. These are people who benefit when you block IL-6, so it would make sense that IL-6 JAK-STAT would be important in these people, because if you block one part of it, their disease goes away. What we weren't expecting to find is that same signaling pathway to be identified as being involved in the people who don't benefit from IL-6 blockade. So we dug into this a bit further. We confirmed that, in fact, in Castleman disease, there is increased activation of this JAK-STAT pathway. So in this increased phosphorylation of STAT3, here are a couple of representative images. And we found there was no difference between responders and non-responders. This suggested to us that maybe you don't need interleukin-6 to activate this pathway in the non-responders, and maybe JAK, which is IL-6 JAK-STAT, maybe if you block JAK in the middle of this pathway, you might be able to control this condition. And so the way we think about it, looking back on our model from before, is that we've shown that IL-6 and JAK-STAT3 are important in some patients, and we've shown that mTOR and potentially secondary to VEGF is important in other patients. Siltuximab can block IL-6, sirolimus can block mTOR. But what this new data suggests is that maybe rather than not knowing which one to block and there potentially being other pathways involved, maybe if we block JAK, which seems to be involved in both STAT3 activation, also mTOR activation, maybe we can stop the disease with something like ruxolitinib that blocks JAK. So there's further research underway. And I think that hopefully this highlights to you how you work to get samples together, get data together, you generate insights, and then you really start digging into what does this mean? What does this mean therapeutically to move from where we were to where we are today? So I'm getting close to being able to wrap up. I just want to highlight where we were in 2012 and where we are today. So over the last 8 years, we've raised about $1.5 million. It's led to about $10 million in additional funding from external groups. We got the first NIH grant to do Castleman's research. There are over 1,200 patients in our registry. We went from there being 2 organizations before us to 3 organizations when we joined or when we came on the block, to now there just being one unified collaborative network where we all work together. There is a biobank now. We've made progress. The questions remain. There is a diagnostic criteria that's been published. There are treatment guidelines. There are predictive biomarkers, like I described, that predict whether you can get better or not on siltuximab. Siltuximab went on to get FDA approval, and now there are 2 drugs in development, both sirolimus, the drug that's saving my life that I'm on every day, and also ruxolitinib, the drug that I mentioned, is now under investigation. So I'm actually going to skip through this pretty quickly just to say that, and to answer the question around COVID, when COVID hit really hard the States in early March, we decided and we realized there's so much overlap between Castleman disease and COVID and our center, which is called the Center for Cytokine Storm Treatment and Laboratory, that we would redirect a large portion of our center towards COVID. And so we launched something called the Corona Project, which you can access the website on the bottom of the screen. And I'm going to go through this quickly just to say that we are tracking all drugs used worldwide to treat COVID-19 in the central database so we can prioritize drugs for randomized controlled trials. I'm going to skip through this to say this is how we do drug repurposing. I'm also going to skip through this. This is one of the ways that we advance research for Castleman disease. Skip all the way through it. This image is trying to highlight that there are about 10,000 known human diseases. And actually, I'm going to take this moment to do something a bit interactive. So if there are about 10,000 known human diseases, would you guys write either into the chat function or into the Q&A, what percentage of human diseases do you think have an FDA-approved therapy? I'll give you guys a minute or so. But if you'll just write it into the chat or Q&A, if there's 10,000 human diseases, what percentage of all human diseases have an approved therapy? FDA has approved a drug to treat that condition. John's come in with 10%. It's a very, very good guess. There's a 5%, 10%, 40%, 11%. You guys are giving some good guesses. 8%, 40%. So I actually, just from this number, the numbers I'm seeing here, the weighted average that you guys are all guessing is about right. So about 25% of all human diseases have an approved drug. I'm glad to see that you guys are on the low end, because that means you realize there are a lot of drugs that don't have anything approved. So I'm going to go back to sharing my screen, and basically it's 25% of all human diseases have an approved drug. 3/4 of human diseases don't have a single approved drug. And when you think just about rare diseases, it's only about 5% of rare diseases that have an approved drug. So we've got a long way to go, and that's why it's so important that you all continue to pursue your pre-health work. So this is a cartoon of an old dog reading a book about new tricks. And the idea here is that I think we need to look at all these old drugs, the drugs approved for the 2,500 conditions, and try to understand which of those drugs may be effective for the 3/4 of human conditions that don't have a treatment, like the drug sirolimus that's saving my life. How many more drugs like sirolimus are there out there? And it's essential that we work together across all players, and that it's not just physicians and researchers working on this, but that they're caregivers and patients and advocates, FDA, pharmaceutical companies, all working together, because there's really just no way that any of us can do this alone. So that brings me to my last slide, which is just some lessons learned. And so the first is that there's incredible power when patients drive research. No one in the medical system is as passionate and as driven as patients are to make progress. I hope all of you will be, and I know you all will be, the next wave of leaders in biomedicine. But I think it's not right for us to believe as physicians or researchers that we can do this alone. We need patients to be a part of this fight. It can't be doctors and researchers paternalistically moving things forward for the patient. We need to partner with our patients to drive forward science. And I promise you it'll move more quickly if you do that. The next is that hope can have many different forms. If I had kept just waiting and hoping that someone somewhere would figure out my disease, I would not be alive today. But it's because I turned my hope into action that I'm alive today. The third is that solutions can be hiding in plain sight. The drug I'm on was at my neighborhood CVS for years while I was in and out of the ICU, but no one had ever thought to try it. How many more drugs are already out there that we need to uncover? The fourth is that we have to systematically capture and integrate data. The insights we gained from my case were only possible because we systematically brought together all the data. We went through it, we integrated it, we analyzed it in ways that would not have been done otherwise. And lastly, it really takes an army. My book's titled Chasing My Cure, but it really should be called Chasing Our Cures, because it really has been such a team effort, and it's really been all hands on deck. So I hope you all will be a part of the Chasing My Cure army. I hope you'll join our army, helping to raise awareness about Castleman disease, about rare diseases, drug repurposing, about Chasing My Cure. And I think we've got time for about 10 more minutes of questions. So thank you guys so much. I should also switch over to this slide to thank my incredible team that has contributed blood, sweat, and tears to our work, many of which are listed here on the screen.
We have a question for you in the Q&A.
Great. Let's see. Do you find that when you block the mTOR pathway, there are many secondary issues related to decreases in protein production or increases in abnormal cell growth by inhibiting autophagy? So it's a great question, Megan. We don't know all of the issues that occur in Castleman disease patients because you've blocked mTOR. We do know that for some people, you block mTOR and their symptoms resolve, that they get better, and there's nothing better than seeing that to see a patient improve. However, there are people who their Castleman disease doesn't get better. And so then we typically stop therapy and we just move on to other drugs. So for the people that their Castleman's gets better, we don't know yet the negative or secondary impact of being on an mTOR inhibitor for Castleman's patients. So we can tell you that we haven't run into many problems of long-term sequelae in the people who are on the drug. One of the nice things about sirolimus is that it was developed for kidney transplantation. So it's meant to be given long-term, daily for years, decades even, patients get sirolimus. So there's a good long-term safety profile. Though, as you mentioned, mTOR is important to a lot of biological processes. So when you start perturbing those things, you certainly are at risk of problems.
Elaine has a question.
Let's see. Oh, nice. Are you able to speak to any tactics you use to juggle and balance everything in your life, from school to research to my own care and my personal life? This is a great question. I'm glad you asked it, Elaine, because it was not easy. When I give this talk, I think it probably maybe sounds like it was easy, and it sounds like there were just only a bunch of highs and no lows, but it was really, really tough. And I worked really, really hard. I know all of you do the same. I mean, for me, I spent most of my weekends of my 20s, or I guess from the time I was 18 to 30 probably, working like all weekend, every weekend, and missing out on a lot of things that my friends were doing, and a lot of time with people that I care about. And so I guess the point here is that for me, it required a lot of work and a lot of focus. As was mentioned earlier, I did a master's at Oxford after undergrad, and then I did medical school at Penn, and then I did an MBA at Wharton. And in parallel, I was running one foundation called AMF for many years before turning over to run the second foundation, the Castleman Disease Collaborative Network. So that's not possible if you don't work all the time. I mean, it's not like I have some sort of magical power where I could, add hours to the day, or just like, work a little bit and get a lot done. I had to work like harder than anyone else I was around. And that's not easy. It's not necessarily something I recommend. I mean, it was hard, but it has to be like, what you want to do. So for me, I think one part of it is that it just takes hard work. And that's not always like an easy answer or easy pill to swallow, because I think that we kind of all wish that there were easier ways. So one part is takes hard work. The second is that I think it also takes incredible focus. You can't work hard on a million things and do a million things well. You can work really hard on a few things and then do them really well. And so for me, I think that I was able to make the progress that I've been able to make against Castleman disease, and also professionally, because I was very, very focused. I worked on a handful of things, and I worked on them really, really well. And some people are better at doing many different things, but I think that it comes down to math, and it's like, it's the amount of time you put into each one of those things, in my opinion, that helps, that leads to progress, or at least correlated with progress. So I think hard work, focus, and then for me, I think the third part I would say is mission. And that's that I didn't do anything that didn't 100% align with my mission and what I'm trying to do. So I didn't do a research project because I wanted to put it on my resume. I didn't do a thing because it seemed like I should do it. I did it because it was part of my mission and what I was trying to do at the time. And so that's not to say that sometimes in life you do things that aren't exactly what you want to do for the rest of your life, but it has to fit within your mission. You don't want to just do a paper to publish it because you feel like you have to. You want to do that paper to publish it because you care about the results, and you think that experience is going to lead on to something in the future. So sorry if that felt like I'm lecturing and I'm on a high horse there, but I think it's an important question. Hard work, focus, and making sure everything you do fits within your mission.
And Pearl has a question.
Sure. So what changes do you think could be made to the structure of medical education to better accommodate students with disabilities and chronic illnesses? I think this is a really important question. I think that going through medical school rotations, if you have a disability or a chronic illness, becomes more complicated. And it really comes down to being able to work with whoever your instructor is to accommodate you. I think it's hard for the system or the university to make too many generalized accommodations, because they really need to be accommodations for you or for what you're going through. So for me, my fourth year of medical school, I was getting chemotherapy infusions every week, and I had a very weak immune system. So I really couldn't afford to be on the really sick units of the hospital. I would have likely gotten very sick myself. And so my preceptors, my instructors at Penn, worked with me to get me on a floor where I was still working the same amount of hours as everyone else, because I could. And when I couldn't, they didn't make me work the same amount of hours. There were times where they made exceptions. But generally, we did smart things to avoid putting me at major risk. And I think it's really, I wish that there could be kind of systemic changes that could make it easier. But I really do think that it's about personalizing the experience to what works best for you. And I think as a result, it makes it harder to generalize too much.
Dr. Fajgenbaum, I was wondering, what challenges, what are the greatest challenges that you've overcome in being able to obtain sustainable funding for your research?
Yeah, so I think that the first challenge to get funding was that there was so little basic kind of fundamental understanding about what was going on that no funding body wanted to fund like, let's figure out what's going on from just the very beginning. I mean, funding bodies like to build upon previous work. They like to know that if they give $1 million for this research, it's going to lead to X, Y, and Z things. But if nothing is known, then it's hard to build upon that. So I spent several years really working on the foundation of things. What is the diagnostic criteria? So we know we're dealing with the same patient population. What are some of the basic understandings from years of research that have never really been synthesized together? So I think getting that fundamental foundation was the first step that you have to do to move forward and get funding. And then I think the second thing is that we took big shots on goal and big swings for the fences, in the sense that we weren't interested in doing a research study to publish a paper. We're interested in doing a research study to lead to a finding that can lead to a drug that can save lives. And I'm not trying to say that other researchers aren't doing the exact same thing, but I think that we, as a result of our kind of long shot, or at least like kind of long-term vision, that meant that we took some risky bets on doing research studies that maybe wouldn't pan out. They either would be, you get either no paper out of it, or you get like a game-changing result that leads to, you know, a really big breakthrough. So we took a lot more risks with that sort of stuff that I think funding bodies appreciated, not because they wanted to fund the risk, but because the risk paid off, and then they wanted to fund moving forward what we found.
Mm-hmm. Mm-hmm. Great, thank you. And Kim has a question.
Sure. Let's see. Take a look at the Q&A. These are great questions, by the way. Kim, how would you say that my MBA experience contributed to the whole process, my medical, my current research, and also my overall perspective? I think my MBA contributed in a lot of ways that maybe sometimes I don't even realize, but I do think that the ways that I do realize it contributes are related to some of the softer side of business, where it's how do you interact with people, how do you negotiate, how do you manage a team, stuff that maybe you wouldn't think would be all that important, but man, is it important when you're trying to get people to collaborate with one another and get people to work together when there are no incentives for that to happen. Being creative and kind of using tools that are not typically in the medical toolkit, but are in the business toolkit, I think have been really, really helpful. And then I think some of the strategic planning that I got from my MBA has been helpful as I thought about kind of coming up with a plan for the field, coming up with a plan for my center. So I think there have been some kind of very tangible benefits, but also some ones that I probably don't notice that I'm benefiting from, but that I think they really are. Now, I don't think that it requires doing an MBA to get those things. I think that the first step is recognizing that they're important, that those skills and those tools are important. And if they are important to you and you are interested in some maybe more systems level impact in healthcare, then I think you can do things to get there without having to go through a 2-year full-time MBA. There are courses you can take, there are books you can read. You don't need the MBA to do it, but for me, the MBA is what got me in that direction.
Right, thank you. And Lucia?
Sure. How do you think your educational path has benefited you as a physician scientist? Do you consider your path to have been non-traditional? Yes, I consider my path very non-traditional. There aren't very many MBA folks that run basic and translational research laboratories. I think that makes me kind of different in a way that maybe others would say that's not a great thing. I think it's different in a good way. I think that diversity of opinion, diversity of perspectives and backgrounds is always good. So I think that my non-traditional route has been helpful for me, but I think I've also recognized that because I'm non-traditional, there are things about my background that are lacking and gaps that I need to make up for, either by self-teaching or by really leaning on great collaborators. And so I think it's about recognizing where our strengths are, where our weaknesses are, and never being too proud or too prideful, and always having humility to recognize that that not one of us can do this alone, and all of us need to work together.
I think we have time for 2 more questions.
Sure. So from Elaine, let's see, you mentioned personalization. Did you have challenges speaking up and being your own advocate when you needed to ask for help or accommodations? I did. I did. I think that I was nervous early on about how people would provide accommodations, or if they would, just how they would treat me. I do have to say that, and every school is going to be different, but I do have to say that at Penn, they were so supportive. The administration, we used to call it Suite 100. It's actually in a different building now, but the administration at Penn Med is really just incredible, and they were so accommodating to me. I mean, they literally, like, people from Suite 100 traveled down to North Carolina to see me when I was in the hospital because they cared that much, and so it was really incredible support that I got. And then the last question, how did you identify people to reach out to help you build the Castleman community in the building, or in the beginning? So the first thing I did was send an email out to all of my medical school classmates, and only one person replied. The email was like, I've got this disease. They all knew I had Castleman's, but I said, I'm going to take this thing on. I need your help. Who's in with me? And only one person replied, but the person who replied to me was doing an MD PhD, and he had done 4 years of really intense research at Harvard before he came to Penn. He's one of the smartest people I've ever met. And so I didn't have, I guess, very much quantity of help early on. It was just me and Chris, but I had incredible quality. And so he helped me to think about the disease in so many ways that I wouldn't have thought of on my own. And it really was Chris Nabel and I against the world early on. But we started making some exciting progress. And once we started publishing papers in major journals and really started shaking up the field, then a bunch of my other classmates came to me and said, I want to get involved too. And pretty soon grad students were involved and business school students. And pretty soon it was literally an army of dozens of people all volunteering their time. And we are always looking for people to be a part of our army. So it's really easy to find my email address. Jackie can give it to you. It's also easy to find online. Email me if you're interested in getting involved in some way. In the past, we used to have work study opportunities, and we used to have a number of pre-health students that were part of post-bacs. We've actually kind of backed away, and we're doing less with work study students, but we do bring on individuals in research capacities. We bring on full-time and part-time. So let me know if you're interested in joining on, and if you're interested in being a volunteer and being involved in a much less formal and much less intense capacity.
And the university has allowed the post-bac students to participate in those kinds of activities, whereas undergrads cannot, at this point during COVID.
I'm glad you brought that up. I didn't realize that.
Yeah. Mm-hmm. And I'm curious to know where we can get a lapel pin. Castleman lapel pin.
It's awesome, isn't it? This is a Castleman. I would be thrilled.
I love it. Do you sell those?
We don't sell them, but we should start selling them.
Good, yeah.
So maybe you and I can connect offline, and we can figure out if
definitely
anyone want to wear this to raise awareness. We would be happy to give you one.
Yeah, I would love one.
But I'd be happy to connect you with someone from the team to figure it out. I love it. One thing you can do other than wearing a lapel, though, is do that warrior flex. So take a picture of you and your classmates in person. I would have everyone stand up and do a warrior flex.
We can do that. We can put that on our website.
Yeah, put it on your website. Post on social media the hashtag warrior flex and hashtag Castleman. Love that. That would mean a lot.
Okay. That's great. So I don't want to take up any more of your time because I know how valuable it is, but I just have to, give you like the biggest, thank you from me and from our students. I really have learned a lot in this time with you, and I really appreciate your being here and sharing your experience with us.
Well, thank you so much for having me. It really was a delight for me, and what awesome questions you guys had. So I hope this was helpful for everyone. And
they're great. They're awesome students. They really are. Thank you.
Thank you all so much.